So I would like to welcome everybody and thank you for joining us for today's webinar entitled Beyond Biogas, How the Waste to Chemicals Alliance is Transforming Anaerobic Digestion. Hosted by the Ontario Water Consortium, we're excited to bring you this session as part of the ongoing work of the Waste to Chemicals project. This is an NSERC Alliance and MITEC-supported initiative led by Professor Christopher Lawson at the University of Toronto. This project is exploring new frontiers in anaerobic digestions, moving beyond traditional biogas production to recover higher value chemicals from organic waste streams. Today's presentations will showcase innovative approaches in microbial engineering, membrane technologies, and process optimization that are shaping the future of resource recovery in Canada. The Waste to Chemicals Alliance brings together key partners including Veolia and Vera, CBS Bioplatforms, and the Ontario Clean Water Agency, as well as membrane technology experts at the University of Toronto. The Ontario Water Consortium is proud to support this groundbreaking collaboration, which exemplifies how academic research, industry, and innovation ecosystems can work together to create cleaner, more sustainable solutions for waste management. We did share the agenda in advance, but here it is again for your convenience. And with that, I will now turn it over to Professor Christopher Lawson to start us off from the University of Toronto to begin the presentation lineup. Chris, the floor is yours, and I will stop sharing.
Great. Thank you so much, Rob, for the introduction. And thanks, everyone, for coming. Can you guys, are my slides? You can see my slides, okay?
We can see your slides. Looks good.
Excellent. Okay. Yeah, so thank you all for coming. Today's presentations will be focused on the idea of making products beyond biogas, really focusing on what we can do with anaerobic digestion for a circular economy. And so when we think about anaerobic digestion, we like to think of it as having kind of a big mouth, right? So it can take in a wide range of different types of feedstocks. It can take in source separated organics. It can take in wastewater biosolids. It can take in renewable biomass. But its product spectrum currently is pretty limited. So it can only make generally biogas, which is a useful product. But really where our project started is thinking about what else could we make beyond biogas? What other kind of products can we make? And so typically when we think about products beyond biogas, you're often faced with, oh, anaerobic digestion is too complicated. We don't have the tools in place to be able to make products beyond biogas. And really empirical methods have been kind of king for tailoring the biology of these processes, which has been a bottleneck. And so in our lab, we're really focused on kind of opening up this black box, understanding the function of the different microbes in this process, but more importantly, really thinking about new approaches for assembling and tailoring the composition of these microbiomes, and importantly, thinking about how we can retool them to make higher value products, which of course is the focus of today. And all of these questions really require new thinking on how one engineers and manipulates the biology of these anaerobic systems. Now, when we think about retooling the biology of anaerobic digestion, we like to think, and toward higher value products, we like to think of this value volume pyramid. And so on the base of the pyramid are energy products. So these are things society needs in kind of the billions of tons per year, right? So we need a lot of them. But their selling price is relatively low. So they're kind of considered lower value, cheaper products. Now, at the top of the value volume pyramid sits things like fine chemicals, pharmaceuticals. We know these things sell for a lot higher selling prices. These are in the orders of thousands of dollars per kilogram, for example. But we've all had COVID injections, for example, and other type of vaccines where we know the volume of the product requires very, very low, right? So with anaerobic digestion, we have a lot of waste, right? We have over 5 billion tons of organic waste being generated across the world globally. Right now, AD is really focused on the energy pyramid, or the base of the pyramid. We want to move it up toward the middle. So really focusing on kind of the bulk chemicals, the plastics, and the polymer space. And so here we'd be able to still put our waste into chemical products to meet our treatment objectives, while also recovering a higher value product from the process. And so this would really expand the economics of anaerobic digestion processes and really allow AD to be more widely adopted across the country and globally. So when we think a bit more about the products we want to make, of course, when we think about potential to offset greenhouse gas emissions, as well as recovering a higher price. So the chemicals market, of course, is the big one, but also things like long haul transportation fuels are certainly in the target. And so these are biofuels, for example, that are involved in aviation and shipping, which are sectors that are generally harder to electrify and really still demand a liquid fuel-based product moving forward. The other thing I wanted to paint here, which I thought this is an interesting article recently from Nature talking about the refinery of the future, is kind of what do the feedstocks of the future look like and what do the products of the future look like? And where does anaerobic digestion fit? So here you can see in 2020, most of the fuel and chemicals we make are coming from oil, right? But also this other bin, which is largely natural gas. And so really catalytic thermal chemical processes are really driving conversion of oil and natural gas to these products. But in 2050, it's really anticipated this refinery to change dramatically, right? And so the feedstocks of the future are really thought of more as CO2, agricultural and municipal waste, right? And the product spectrum is shifting more toward chemicals and polymers, given that the demand for carbon-based fuels is really shifting toward non-carbon-based sources, things like solar, wind, energy, hydropower. And so really what we'd like to think of here is, could we put anaerobic digestion as a key platform technology, helping us transform all of our carbon-based waste feedstocks into the polymers and chemicals that we'll need in the future. And really one of the key points here is that, waste contains a high amount of carbon, right? And so if we can put that carbon into products that requires carbon, it'd be advantageous, especially given that the energy sector, again, is really moving away from carbon-based fuel sources. Okay, and so then what should we be making? What's a good starting point? So our lab is focused really on making medium chain fatty acids, which we think are a great starting point for waste to chemical biomanufacturing. MCFAs themselves are a lot more valuable than biogas and methane, so their selling price is around $4 to $5 a kilogram, and they're used in a wide range of applications. So you'll see them in nutraceuticals, for example, on your shelf. You can go to Shoppers Drug Mart. You'll see them in personal care products. This is beard butter, for example. Been working on my beard a little bit lately. One of the main products, though, especially in North America, is for animal feed applications. So medium chain fatty acids themselves are antimicrobials. And so what the livestock industry has been using them for rapidly, which is increasing, is as an alternative to antibiotics. Right now, so the MCFAs have a high selling price. They have a wide range of applications they're used in. And one of the other main reasons to start with them is that current production comes from palm and coconut oil refining. And I'm sure you've all heard about the issues with palm and coconut oil refining. I've listed some of them here. They include, large greenhouse gas emissions, obviously deforestation, as the picture alludes to, biodiversity loss, soil degradation. But beyond these environmental issues with palm and coconut oil refining, a lot of the supply chain for bringing MCFAs overseas, for example, to North America, are really long and nuanced and volatile. So it requires, taking the trees themselves and extracting oils from them in Indonesia, that then gets processed, for example, in the Netherlands, and then that eventually makes its way over to North America. So the supply chain is long and quite complicated. And because of this, the volatility in the price of MCFAs fluctuates, which can impact people who actually use these products. And of course, just the logistics of getting them over to Canada can be very complicated. So there's really a strong demand on the consumer side to have more locally sourced, environmentally friendly methods of making these MCFAs. So as I mentioned, MCFAs themselves are valuable directly for application as an animal feed additive, for example. But the other nice thing about them is they're a bulk chemical. So they can be rapidly made into derivative products. For example, alcohols, alkanes. Alkanes, for example, would be the precursor or the chemical being used in a sustainable aviation fuel. They can also be carboxylated to make things like adipic acid. These are precursors for nylon. And there's existing chemical methods already in place to be able to do much of the chemistry involved here. But also, certainly in our lab, we're very interested in doing this via biological engineering. And so, of course, being able to make a whole suite of medium chain oleochemicals from our waste really would broaden the market size. Each one of these products has market sizes in the billions of dollars. So this would really enable a bio-based oleochemical production from waste refineries. And so this really leads us to our project here. And so really this simple question that the project asked was, can we convert source separated organic waste from the city of Toronto into medium chain fatty acids? And so this, as Rob mentioned, was an NSERC Alliance project. And so what we did it in a big picture is we went out and we collected source separate organic waste from the local facilities here in Toronto. And we first started to develop the bioprocess knowledge. So how to operate reactors, how to separate out MCFAs from the fermentation broth, and develop and demonstrate systems for being able to do this with real waste. So you'll hear a number of talks about that today. And the other piece we really focused on was laying the groundwork for really developing microbial communities in a more bottom-up fashion that would also allow us to genetically manipulate some of these microbes to make some of the other higher value products that we want to access in the oleochemical space. And so you'll certainly hear some talks about that as well today. And so with that being said, I'd like to pass it on now to Jasmine, who will be taking you through some of the work on our bioreactors. Thanks.
Okay, thanks, Chris. So first I'll start off with traditional AD to picture this study. So there are certain municipalities, such as the City of Toronto, which collect and treat source-separated organic waste, which is basically the waste that we throw in our green bins, through anaerobic digestion as a waste diversion strategy. And so this process involves a diverse set of microbes which break down the complex polymeric material into monomers, intermediate acids and alcohols, and then finally biogas. And then this can eventually be upgraded to RNG. But by driving the microbial process away from methane production, we have the potential to drive higher value products by using the substrates that were initially produced in the earlier stages of AD. And so the key substrates that we're interested in for our process to make MCFAs are lactate and ethanol, as well as amino acids and glycerol. So the organic waste that we use for the study is sourced from two organics processing facilities in the City of Toronto, Dufferin Organics and Disco Roads. And both of these facilities basically collect SSO waste from households and public buildings, and then they pre-process it to remove inorganic material. And then they add processed water to the organic fraction to form a pulp that can be pumped. And then this is what is fed to the digesters to produce biogas that is upgraded to RNG. And so generally, these two facilities have very similar designs. The main difference that we found is that they use different equipment in their organic separation. So Dufferin uses an organics extrusion press, whereas Disco Rose uses a BTA hydromechanical treatment. But regardless of this, the important aspect is that the waste that we're feeding into our reactor is this liquid slurry SSO pulp. And this is the feedstock for the MCFA production. So the aim of our study is to demonstrate continuous MCFA production and recovery from SSO waste in a bench scale bioreactor. And the way that we're doing this is through an open culture fermentation approach. And this means that we're using the existing microbial community from the waste. And by controlling the environmental parameters in the reactor, we can enrich the microbes that are responsible for MCFA production. And so we've been operating this reactor for over 2 years now, anaerobically at 37 degrees Celsius and a pH of 5 to inhibit methanogenesis. And we've collected SSO waste as our feedstock from the Dufferin facility for the first 441 days. And then when they started having some process issues, we switched to collecting pulp from the Disco Roads facility. And I also want to highlight that this pulp is particularly well suited for our application since it contains substrates, particularly lactate, which are important to produce MCFAs through a process called chain elongation. So we first investigated what would be the impact of adjusting solid retention time of 8 days versus 12 days on titer rate and yield metrics in our reactor. And we basically found that increasing the SRT resulted in a higher MCFA titer and yield when we compare the best performance in terms of a 5-day average. However, we found that the production rate did not improve that significantly. So here we'll take a closer look at the volatile fatty acid and lactate profiles at the 12-day SRT, particularly because we operated at this period for a very long time, over a year. So we are able to account for seasonal variations as well as track the facility-specific variations of the pulp and see how that actually impacts the reactor performance. So we found that the changes in the outdoor seasonal temperature influenced the lactate concentration in the SSO pulp when we went and got to go and collect it from the facility. So we first observed the highest lactate concentration when we collected SSO pulp from in the wintertime. And then during that time, we also achieved the highest hexanoic and octanoic titers and MCFA yield. And then when we see that it got warmer during the spring and summer periods, the lactate concentration decreased, and then this corresponded to a decrease in the MCFA titer. And just from this process data alone, we can see that we're getting some sort of insights on how the feed variability impacts the reactor performance. However, when we actually compare the MCFA yield to the MCFA titers, we find that the yield is not affected too much by the seasonal lactate variation. And this is likely due to the fact that during the periods where the lactate is quite low, there are other substrates that are available in the SSO pulp, such as amino acids, which are being used to make the MCFAs. However, we still have the potential to increase the yield by continuously extracting toxic MCFAs from the reactor. And this is something Diana will go and expand upon later in this session. Additionally, I wanted to mention that we can see the effects of the feed variability due to the seasons on the MCFA product selectivity. And we can see this using a COD balance. And we basically see that in the winter, the high lactate in the feed contributes to more even chain fatty acids, such as hexanoic acid, which is shown in the dark red. Whereas in the fall, most of the lactate in the feed has already been degraded to short chain fatty acids before we even got it to our lab. And it was mainly degraded into propanoic acid. So this led to an increase in odd chain fatty acids in our reactor. And so next we wanted to transition from the process level MCFA production to exploring how these operating conditions impact the microbial community dynamics. So we investigated the dynamics using full-length 16S rRNA sequencing with the Oxford Nanopore technology that we have at U of T. And we basically analyzed the microbial composition in the SSO feedstock and also looking at the time series of the bioreactor community throughout all of these different process conditions that we tested. So in the sequencing work, it was in collaboration with Dr. Blake Lindler, and we first examined here the microbial composition of the SSO feed collected from the two organics facilities. And what we see here is that lactic acid bacteria were dominant in both of the SSO feeds from both facilities, as previously shown. And this is consistent with the high lactate concentration. And although the two processing facilities both receive the same green bin waste from the City of Toronto, the dominant lactic acid bacteria genus differs. So for example, Dufferin is dominated by Lactobacillus, whereas Disco is dominated by Streptococcus. And this basically just suggests that the local environment and the processing conditions are driving this site-specific microbial selection. And now when we'll take a closer look at the microbial community dynamics in the bioreactor over our time series, and we see that the reactor microbial profile is dominated by lactic acid bacteria, carried over from the SSO pulp that we were feeding the reactor. However, another dominant functional guild found in the bioreactor are the chain elongating bacteria that are involved in MCFA production. And this includes a novel eubacterium and a Pseudoramobacter genera. And then at first glance, this sequencing data suggests that switching the feedstock from Dufferin to Disco had a significant impact on the microbial community profile, showing more diversity after we made that switch. So here in this plot, we are seeing how similar the microbial communities are in the feed and reactor samples to each other based on how they cluster together. So the closer they cluster together, the more similar they are. And we see that the reactor sample points across the different operating conditions occupy the same space relatively closely. However, when we look at the feedstock from Dufferin and Disco, there are distinct clusters. So the separation along these types of feedstocks suggests that switching the feedstock had a significant impact on the microbial community composition than when we were just looking at the changes in the reactor operation alone in terms of like the volatile fatty acids and lactate. And then finally, as an exploratory analysis, we did preliminary correlations on the top taxa with process data including lactate, short-chain fatty acids, and medium-chain fatty acid concentration profiles in the bioreactor. So we found that lactic acid bacteria show a positive correlation with lactate, with also some correlating with short-chain fatty acids, which suggests that both homofermentive and heterofermentive metabolism is seen here. And we also see two putative chain elongating bacteria, Eubacterium and Pseudomobacter, partly correlating with MCFA production. And then to further investigate the microbial functional activity, the next steps that we are going to be doing is including the studying of metabolism of isolated strains from the reactor and the feed and doing more deeper omics analysis. And that's the end of my presentation, and I'll send it off to Ethan.
Thanks, Jasmine. Can everyone see my screen?
Yes, we can see it.
Great.
It's just not in presentation mode right now. Oh, there we go.
Okay, great.
Now it's
so
awesome.
Hi, everyone. I'll be presenting my presentation about engineering microbial communities to expand anaerobic fermentation to new products within synthetic biology. So I will be describing two main approaches that we conduct in our lab, which includes synthetic community engineering and metabolic engineering. So to begin, there are two main approaches for microbial fermentation. Jasmine described something called a mixed community approach, where the organisms arise from the feedstocks themselves. And the benefits of this particular approach is that you don't need to sterilize your feedstock, and you can enrich microbes on any given organic waste stream. But the drawback of this approach is that it can lead to lower selectivity for your product that you desire, such as MCFAs, because there might be competing microbes that also compete for the same substrates. So another approach is something called synthetic communities, where you take only the microbes with your desired function, you combine them into a synthetic community and use them in your bioprocess instead. And this is beneficial because you're able to achieve higher selectivity because you've only included microbes with the desired function. But this might lead to potential hydrolytic limitations, especially with more complex organic wastes. But towards this approach, our lab has been developing a systematic way of engineering these synthetic communities with the DBTL cycle, or the design, build, test, learn cycle. And how this works is that you first source organisms from different contexts, such as the cow microbiome, or the MCFA bioreactor, and through growing them on organic wastes, you're able to obtain these stable, self-assembled microbial communities. The next step is to take these microbial communities and split them into the different microbes that make them up. And then you are able to characterize these microbes into different functional guilds, such as the hydrolytic bacteria or the MCFA-producing bacteria. And through an automated microbiome assembly pipeline that we're developing and the DBTL cycle, we're able to recombine these organisms into synthetic microbiomes with a certain desired function, in this case, MCFA production. And then on top of this microbiome engineering, we're also able to take the isolated strains themselves and modify them on the genetic level to enable an expansion of their capabilities. And ultimately, what this process allows us to do is boost product titer rates and yields, link the microbiology to the bioprocess at large, and also allow us to make higher value chemicals. So for the first piece, sourcing the microbes, Joel Howard, a student in our lab, in collaboration with Professor Wang at Columbia University, was able to use an automated system to isolate over 2,000 strains on over 200 different conditions. So using the system, we were able to generate a biobank of different microbes isolated from municipal waste, anaerobic digesters, and also our MCFA bioreactor. Now diving a bit more deeper into the DBTL cycle. From the biobank, we were able to characterize the different functional guilds of these different isolates into MCFA-producing bacteria and also the hydrolytic bacteria. and propose different community designs that have these different microbes in different combinations. In the building phase, we are leveraging an anaerobic biofoundry, which I'll describe a little bit later, to construct well plate assays with the different community designs and grow them in food waste media. With our system, we're able to grow and measure the MCFA production for these different community designs. And this information allows us to propose new designs using machine learning and Bayesian optimization guided design to propose a new set of communities for the next cycle. So through iterating through the cycle, we're able to incrementally achieve better and better MCFA productivity. So in our lab at U of T, we've set up an anaerobic biofoundry that really helps accelerate this type of research. What you're seeing here is our anaerobic chamber, in which we have a liquid handling robot, which allows us to automate the construction of the different experimental conditions. And we also have this anaerobic incubator, which allows us to test up to 8 experiments at once. So now here's our foundry in action. The video on the left here is showing our liquid handler combining the different strains from the source plate into an experimental plate where the bacteria will be incubated. The next video is showing that once the plate is constructed, our incubator is able to take them and grow them under humidity and temperature controlled conditions. And we can measure up to 8 conditions in parallel. And this system allows us to screen up to 1,000 different experiments per week. So it really increases and accelerates the work beyond what is capable if we were to do it manually on the bench. So after 2 cycles of the DBTL cycle, we were able to really increase the MCFA productivity using our synthetic community designs. The plot you're seeing here is the results of the first cycle where we're measuring MCFA concentration, and it's a random distribution or random configurations of the different strains. But after 1 cycle, after learning from that first iteration, most of the community designs are actually able to improve upon the initial MCFA productivity. And the plot on the right is showing the top 25 conditions where we can see different combinations of the different functional guilds are able to achieve different MCFA productivities. So this has been really a great way of accelerating and screening different conditions that improve upon our open culture technology or the mixed community strategy. Now I'm going to switch a bit more to the engineering of the microbes themselves. And I'm going to be taking you through an analogy here. So engineering bacteria for new functions is much like loading a CD into a stereo. Your CD encodes for all the different songs that you want to play, and you can insert this into the stereo in order to play your favorite song. And much in the same way, we use plasmids, which are circular pieces of DNA that encode new functions. And once we deliver them into the chain elongating bacteria, we can enable the production of different bioproducts. And through building a kit of different plasmids, we're able to access different bioproducts that these bacteria cannot natively or naturally produce. So I'm showing here the work that we've done so far. We devised a pipeline that allows us to deliver plasmid DNA into these elongating bacteria, which is quite difficult because the strains are quite resistant to doing this. So we did some preliminary engineering to allow this workflow to be really efficient. And now I'm showing here, with this plasmid DNA that we inserted into the strain, we were able to generate engineered bacteria that can fluoresce, which is a really important research tool. And this is the first demonstration of doing this in the strain Clostridium cliveri. So we're really excited about this work because now we can actually change the function of a given bacteria. We've so far reported this work as a preprint on bioRxiv. So if you're interested in more of the details, you can please check that out. But using this system, our further goal is to actually modify the inherent metabolic pathways of these bacteria to expand the product spectrum of chain elongation. So we're currently building a plasmid toolkit with different functions that have been encoded into these plasmids. And with this system, we want to engineer different chain elongating bacteria For example, some that use lactate as a substrate, or some that use ethanol as a substrate, in order to access a wide range of oleochemicals. The first targets that we're going to go after are longer chain fatty acids, which are more valuable compounds themselves, and also fatty alcohols, which are widely used as solvents in the chemical industry and can be derivatized to sustainable aviation fuels. So to summarize, engineered synthetic communities can help improve productivity of MCFA production and also expand the product range of chain elongation. And we're doing this to sourcing different microbes from different contexts, splitting them, characterizing them into different functional guilds, and on top of that, modifying the strains themselves, and then recombining them into synthetic communities that can allow us eventually to access these different product classes. All right, so that's the end of my presentation.
Thank you very much, Ethan. This is the point where we move over to Professor Jay Werber, who's going to talk about unlocking value from waste. Jay? You're still muted.
Jay, you're muted.
Hi, everyone. Can you hear me now?
Yep, louder and clear.
Awesome. Okay, so I'm guessing you can see my slides. Okay, so before I get started on the talk on this actual project, I thought I would introduce the lab that I run. So my lab's called the Advanced Membranes Lab. I started at U of T the same time as Chris, and Chris wrote me into this work, which has been really fun. But since I haven't met most of you, I thought I'd just give a really brief intro into what the lab does. So our goal is to address major sustainability challenges through innovations in membrane materials and processes. We particularly focus on making better membranes. That's a big goal of the group, using polymer science-based approaches. We have a lot of experience with RO, so reverse osmosis for desalination and water treatment, and electrodialysis as well, so using an electric field to drive ion separations. The main research areas in the group are focusing on brine treatment. So really, really high salinity brines is one of the major remaining challenges in the desalination space. And then we're focusing on selective separations of metal ions using monovalent selective electrodialysis, bipolar membrane electrodialysis, and then some carrier-based membrane technologies too, which is a little more niche. And then the last research area is of course what I'm talking about today, which is the purification of bio-derived chemicals, which is really led by the really great work that you can see that Chris's group is doing. Okay, so talking with Chris about sort of what they would need from an MCFA purification process, there were two sort of possible final products that we might be able to produce. The first is a mixed sort of oil of a mixture of MCFAs. So in other words, having predominantly C6 to C12, a relatively small amount of C1 to C6 in a neutral acidic form. And so this could be used in various applications as a mixture. Animal feed, for example, would be able to use this product. The other option is producing sort of isolated, purified MCFAs. In other words, you would have a separation process that would produce pure C6, pure C7, pure C8 as individual products that would be separate. In terms of desired process characteristics, of course, low cost is the biggest one. Second, though, is low carbon footprint, particularly if you can run it on electricity, then you can run it using renewable electricity, since solar and wind, again, are becoming the dominant sources of electricity. Another really important thing is having inline MCFA removal, as Jasmine alluded to, and as Deanna's going to talk about in the next talk. Having inline removal of MCFAs is really important because these are toxic. These are toxic chemicals. And so if we can decrease the amount of MCFA in the bioreactor, this can help produce more MCFAs microbially. We additionally wanted it to be solvent-free. Solvent can gunk up your system. It can also be toxic to the cells. So if we can have a solvent-free process, that would be great. And when I say solvent, I'm talking about organic solvents. And then, of course, we want a robust process. So our initial focus is on the relatively easy sort of final product, which is a mixed MCFA oil. If we were to try to do sort of isolated purified MCFAs, you would likely need to have several distillation steps, which of course is is complicated by energy and cost. So just a little background. I mean, we're not the only folks working in the VFA, MCFA space in terms of anaerobic digestion to produce these bioderived chemicals. One of the big groups that's been looking at this is the National Renewable Energy Laboratory in the USA, so NREL. And the technology that they're really exploring for the separation process is just the traditional process, what a typical chemical engineer would sort of give you as the best process for producing one of these chemicals. And this would be basically using liquid-liquid extraction, where you take an organic solvent. So they're showing various examples on the right. So the one is mineral oil in black. So that's one possibility. And then you have various other solvents that are a little bit more polar, as well as some sort of co-solvents like this one called topo, which can help pull some of the carboxylic acids into the oil phase. And so these work based on solubility. So hexanoic acid, octanoic acid, these are pretty hydrophobic species. And so they're going to dissolve, they're going to partition pretty selectively into this oil phase. Now you have this oil phase that's enriched in these organic acids. You bring that to a distillation, where the distillation will evaporate off, so vaporize off all of these acids. And then the liquid phase from the distillation will be the really high boiling solvent that would go back to the liquid-liquid extraction. So that's what NREL's sort of exploring. They've done a lot of techno-economic analyses and so on, as well as experimental exploration of this relatively traditional process. And it works. This would work well. The issue, though, is that distillation is one of the dominant chemical engineering separation tools. It's effective, but it uses an enormous amount of energy. And also, when you're designing one of these plants, there's a lot of steel in the ground, right? You build these giant distillation columns. They're not very flexible in terms of if you have capacity changes, the need to change your plant to a larger size, for example. And so the process that we sort of came up with is, I would argue more sustainable and one that you can drive using electricity. And so the process is outlined here. So on the left, you have the bioreactor, which Jasmine showed earlier in terms of the performance. And in the bioreactor, you would have submerged ultrafiltration membranes. These ultrafiltration membranes would retain the microbes. And then the permeate would just be a liquid solution that would go to the first step, which is just to remove CO2. And the reason why we're removing CO2 is not immediately evident, but basically it affects the downstream processes. But this is a very simple thing. You just flow the solution through a silicone or PDMS membrane, you pull a vacuum, and this process is selective for evaporating, for pulling out CO2 that's dissolved in the solution. So then the solution goes to the inline extraction cycle. And so this inline extraction cycle uses silicone-based or PDMS membranes and a high pH solution that will pull across the carboxylic acids. And I'm going to talk more about this step in the next few slides. And so this is the main process to extract the MCFAs. The MCFAs are then extracted, and the remaining solution is returned back to the bioreactor. And this solution is rich in the short-chain fatty acids, in alcohols, and things that are not pulled across into that high pH solution. So we run this process for a while. We generate a high concentration of MCFAs as carboxylates in the high pH solution. And then we bring this to the last step, which is called bipolar membrane electrodialysis. This is a commercial technology that's used to split salts into acids and bases. And so using this step, we can generate the acid, which in our case is going to be the MCFA oil. So we're basically neutralizing the carboxylic acids from the carboxylate form back to the acidic neutral acid form. These things are hydrophobic, and so they'll phase separate, form an oil phase. That'll be our final product. And we'll pull across to form sodium hydroxide, which we'll use back in the extraction step. I'm not going to talk more about bipolar membrane electrodialysis because Renzo's going to talk about it in an upcoming presentation. But I will focus on this inline MCFA extraction step, which is the main thing that we've been focusing on over the last year or two. This work has been led by Jiahao Zhu, as well as Renzo, who's talking in a few minutes. And again, the main thing that we're focusing on is using solvent-free polymeric membranes that are relatively easy to make, and also things that you can find commercially to affect this separation. And so these membranes are comprising silicone, which is also known as polydimethylsiloxane, or PDMS. This is a really common polymeric material that's used in membrane science. It's also what you use to caulk, your bathtubs. It's a really common material, right? This is just straight silicone. And the really nice thing about silicone is it's kind of liquid-like in its behavior. The polymer chains have a lot of thermal fluctuation, so they sort of behave almost as if they were a slab of oil. And so you can sort of think of one of these membranes as being a slab of oil, where you have your solution that's from the bioreactor. It's at pH 5.5, relatively low pH. So some of these acids are available in their neutral acidic form. The neutral acid form can then dissolve into the PDMS, into the silicone. The silicone is really, really hydrophobic. So the charged state, the carboxylates, these ionic forms are not going to dissolve into the membrane at all. Only the neutral acid form goes in. This neutral acid then basically diffuses through the polymeric matrix to the other side. And as soon as it reaches the other side, because the other side's at pH 9, 10, or 11, way, way above the pKa of the carboxylic acid, it immediately does an acid-base reaction to form the ionic form, the carboxylate. And so this basically maintains a driving force for the carboxylic acids to diffuse from the left to the right. And we can sort of model this using what we call the solution diffusion model, which basically says that it dissolves in and then it diffuses through, where the dissolution we can define using what's called the sorption coefficient, basically how much does a particular chemical species dissolve in. And then D is the diffusion coefficient, sort of how fast would it move through this membrane material. And the experiments that we do are basically forming flat sheets of a model PDMS film and then running it using in a diffusion cell where we separate this low pH solution from a high pH strip solution. Yeah, Robert?
It was just we're approaching your 11:45. Just a 1 minute warning for your
Great, thanks. So you can see that the process here is very selective for the more hydrophobic species. So C8, the octanoic acid, is depleted almost immediately. C6 is depleted a little more slowly, and then C2 is depleted the least. And when we model this, we find that the permeability, basically the C8, the octanoic acid, is like 100-fold more permeable than the acetic acid. And this is driven almost entirely by how much these chemicals dissolve into the PDMS film. They all diffuse at about the same rates. So basically, in a nutshell, these PDMS membranes enable rapid, selective, and solvent-free recovery of MCFAs into an alkaline solution. And this selectivity is driven almost entirely by solubility, and the selectivity is nearly identical to what you would have with your conventional liquid-liquid extraction, except now that we have a solvent-free system, which is nice. And the other really nice thing is we can use existing commercial membranes. So there's two different companies that make what are called hollow fibers. These are just tubes, so really, really thin straws that can separate these two different liquid phases. And so we can use these membranes directly into our system, which Deanna's going to talk about next. And that is it. Thanks, everyone.
Thank you, Jay.
So thank you, Professor Orber. Good afternoon, everyone. I'll be continuing providing further updates on the bench scale bioreactor study, now with an inline product extraction system. From studying the impact of solid retention time of 8 days and 12 days, we can see that the performance metrics such as rate and yield have further room for improvement. Therefore, the next step of our study was to investigate the impact of continuous MCFA extraction on the bioreactor performance metrics. And now I'll just reiterate some of the things Professor Warber mentioned in this process flow. The goal of the extraction system is to, again, increase the MCFA production metrics by continuously recovering the undissociated or protonated MCFAs from the bioreactor, as they exhibit toxicity to microbial activity. And the extraction of MCFAs here is driven by pH gradient between the bioreactor at pH of 5 and alkaline solution pH of 9, and MCFA concentration gradient. We are using PDMS membranes to achieve selective separation of medium chain fatty acids over short chain fatty acids. However, since the bioreactor has high concentrations of suspended solids, they need to be removed to avoid rapid fouling of these PDMS membranes. Therefore, we're using the solid-liquid separation by using the ultrafiltration membrane submerged in the bioreactor. And this membrane bioreactor configuration will also allow us to operate at longer salt retention times to potentially improve the hydrolysis rates. And the last step of this process is to obtain MCFA product as purified oil, which is done using bipolar membrane electrodialysis, and Renzo will go in more details about this on the future slide. And in the next slides, I'll be focusing on the first two stages of the process. So for the first stage, solid-liquid separation, we're using violet ultrafiltration membranes assembled in 3D printed modules and directly submerged in the bioreactor. And currently, there are 2 ultrafiltration membranes in the bioreactor. The goal of this step is to produce solids-free permeate from high solids reactor broth, which then goes to the PDMS membrane for MCFA extraction. And here we'll take a look at the current membrane bioreactor setup integrated with the inline extraction system. So here we have our feed, source separated organics, pulp, and biomass waste streams. The 2 ultrafiltration modules are submerged in the bioreactor, from which the permeate flows to the 3 PDMS membranes in series from the tube side, and on the shell side, in the alkaline solution at pH of 9 selectively extracts the protonated medium chain fatty acids. And the retentate on the top with remaining short chain fatty acids is being sent back to the bioreactor. And now we'll take a closer look at details of this MCFA extraction step. As we are operating the bioreactor at pH of 5, which is close to the pKa values of MCFAs, approximately half of them exist in their protonated or inhibiting form. Therefore, we want to reduce this inhibitory concentration of MCFAs in the bioreactor. And as those protonated MCFAs exit in the permeate stream of the membrane bioreactor, they are extracted into sodium hydroxide alkaline solution, forming MCFA salt. And the driving forces here are MCFA concentration gradient again, and the pH gradient. And this MCFA salt then can be sent to the bipolar membrane electrodialysis step for further purification as oil. And now we'll take a look at the performance of the PDMS membrane for MCFA extraction. On this graph, we're looking at volatile fatty acids profiles in the alkaline bottle that is accumulating them. And the selectivity of PDMS membranes is promising, as you can see here, reaching more than 80% of medium chain fatty acids over the total volatile fatty acids in that alkaline bottle. The MCFA extraction efficiency originally was quite low, but with the increase in ultrafiltration and PDMS membrane areas, it significantly improved, reaching about 75%. And as we increased the ultrafiltration membrane areas, we also implemented regular backpulsing in place, helping us maintaining relatively steady permeate flow rate, as you can see in the black dashed line. And as the extraction was implemented in place, the MCFA titer in the bioreactor decreased by around 2-fold. And this is the ultimate goal of the extraction, to decrease the concentration of MCFAs below their inhibitory levels. And here we hypothesize that reducing the MCFA concentration will increase the overall performance metrics such as rate and yield. So in the bioreactor integrated with extraction system, MCFAs that are produced exit in two streams, first biomass waste stream and the ones that are pooled in the PDMS membrane step, and taken together, MCFA yield in the bioreactor has reached 0.3 grams of MCFAs per gram of volatile solids in the feed, which compared to the performance without the extraction has improved almost twofold. And the overall yield, which is the product of extraction efficiency and the bioreactor yield, it reached 0.23 grams of MCFAs per gram VS. And this value can be further optimized to reach that bioreactor yield of 0.3 grams of MCFAs per gram VS by improving the extraction efficiency of the system. And this, I showed previously, this can be done by increasing the membrane areas. And these results together demonstrate that reducing the inhibitory MCFA concentration via continuous product extraction can significantly improve the MCFA production performance metrics. And in conclusion, we've listed some of the optimization points for the presented process. So from the solid-liquid separation side, there is an opportunity to operate at longer solid retention time to promote greater hydrolysis and access those unavailable yet substrates to improve the MCFA yield. From the inline MCFA extraction, there's opportunity to increase the extraction efficiency by simply increasing the membrane areas, and that will help us to improve the overall MCFA yield. And also, as we increase the extraction rate, we can further reduce the concentration of the MCFAs in the bioreactor, subsequently improving the bioreactor yield and the production rate. And this is the end of my presentation. I'll pass to Renzo to talk more in details about the bipolar membrane electrodialysis.
Great. Thank you, Deanna. I'm just going to share my screen. All right. Can everyone see my screen? All right, great.
Good.
Great. Yeah, so you guys have heard all the great work that's been going on in terms of optimizing the current bioreactor process, and also Deanna gave a quick update, or a detailed update of their current inline MCFA extraction using PDMS membranes. And really the last sort of step or goal challenge that we have in this project is to be able to regenerate these MCFAs back into their pure acidic form in order to obtain an extracted MCFA oil that we could sell. So going from bipolar membrane electrodialysis at scale, this would be ran in batches. And I also wanted to reiterate, as we run these in batches, the MCFA concentrate would be the accumulation of MCFAs post-PDMS extraction until they reach high enough concentrations for us to regenerate them back into their oil form. So my sort of role in this is that I've been running some bench scale bipolar membrane electrodialysis experiments, or BMED batch tests. And essentially how this works at the bench scale is that we have our MCFA concentrate. This is recirculated through our BMED system. And as this happens, the pH of the MCFA concentrate solution is lowered. And we hope to reach around a pH lower than 5 to ensure that all the MCFAs have been fully converted back to their acidic form. Now, at the same time, we also have another stream here of water, which regenerates this alkaline solution during the BMED loop. So how this works, sort of zooming in on the actual BMED system, what we run is called a two-compartment BMED system because of the fact that we have two streams. One is our MCFA concentrate and one is our water stream. Now, what happens is as the MCFAs are pumped through their own compartment, what happens is that they combine with hydrogen ions that are produced by a water dissociation reaction that occurs at the bipolar membranes. Now, at the same time, we have sodium ions from our MCFA concentrate solution that permeate through what we call a cation exchange membrane. And then these sodium ions combine with the hydroxide ions, which are also formed at the bipolar membrane to create sodium hydroxide. So as we are circulating both of these streams through, we are both regenerating our MCFAs back to their neutral acid form. And at the same time, we are also producing a high pH base stream from our water stream too. We also have what we call rinse solutions that we pump through both ends of our BMED system that allows for current to be carried throughout the compartment. And this can sort of be repeated into what we call n number of stacks, where we could have multiple membranes in line together to help increase the efficiency of our system. So up to date, we ran two bench scale tests, one with a synthetic MCFA solution, which mimics sort of the same conditions that they're seeing at the bench scale bioreactor process. And we also took a real MCFA alkaline solution from their bench scale process to test as well. Now, going through some highlighted results. So what I wanted to focus on was the pH of our solution. So as I said before, we want the pH of our MCFA solution to be less than 5 to ensure that all of them are converted back to their neutral acid form. So the results that I'm showing here for both the synthetic and the real MCFA runs, if you look at the orange lines, what we can see is that the synthetic MCFAs reached around a pH of 5, while our real MCFAs, we didn't see a big drop in the pH, both of them starting around 9 and hitting about a pH of 7 for the real MCFAs. So the reason for this, we know that we had issues with our synthetic solution because of the fact that the water stream, which very quickly in our experiments reaches up to a high pH of around 9, was leaking into our MCFA solution, which sort of decreases the efficiency that we were hoping to achieve. So that sort of limited how much we could drop the pH by. But the more prevalent issues, of course, with our real MCFA solution is due to biogas formation. So in the bioreactor there's the anaerobic digestion process, which produces a lot of biogas or CO2. As the effluent leaves the bioreactor around a pH of 5 or 5.5, the mole fraction of CO2 mainly exists as CO2 itself. But what's happening is that as we are extracting the MCFAs using the PDMS membranes, CO2 is also co-extracted. And as they encounter this alkaline solution that the MCFAs are in, they are converted into their carbonate and bicarbonate ion forms. Now, this poses a really large issue when it comes to processing or comes to regenerating the MCFAs using our BMED system, because you can imagine we have more of these negatively charged ions in our solution. As these are pumped through the same BMED system that we have, they sort of compete with the MCFAs for these hydrogen ions, which sort of creates this buffering effect. And because of that, we weren't able to bring the pH down to lower than 5 as we would hope. So as you can see on the left as well, you can qualitatively see the formation of these bubbles in solution as we're regenerating the MCFAs, trying to drop that pH where the CO2 comes back into solution and is sort of bubbling out. So talking with Professor Lawson's team, we sort of concluded that CO2 removal is required for PDMS extraction to sort of limit this buffering effect that we're seeing and help increase the efficiency of our BMED system. So Yeah, so this step would be implemented prior to inline MCFA extraction with PDMS membranes. And of course, we ran some bench scale testing as well, using the same PDMS membranes that we use in inline MCFA extraction. So what I did at the bench scale is that I had a synthetic solution that I bubbled with CO2. I pumped that through one of our PDS membrane modules, while at the same time pulling a vacuum on the shell side of the membranes. And then from there, the first thing that we wanted to do is to see if we were actually extracting any of our MCFAs from this feed solution with the vacuum being applied. So first we looked at the concentrations of the MCFAs and other volatile fatty acids going from the synthetic feed solution to the raffinate. And as you can see, we have a relatively stable concentration of MCFAs going from the feed to the raffinate, with the exception of C8, which we expect is sort of dissolving into the PDMS membrane, which is why we see this sort of discrepancy or this decrease in the concentration. But then again, these tests are ran at very small volumes. So at a much larger scale with the bioreactor, we might not see this much of a drop in C8 concentration during the CO2 removal step. Now, the other thing that we looked at, of course, was the amount of dissolved CO2 in both the feed solution and the raffinate. So we saw from our testing that we were able to remove around 67% of CO2 with just one PDMS membrane module. And we sort of theorized that two of these modules in series could help remove up to 90% of CO2. And just to summarize what I've talked about in terms of what we've been doing for MCFA purification, with our BMED system, we didn't really observe any sort of emulsification of the MCFA oil and the aqueous phases as we were running our tests. We sort of showed a proof of concept that we are able to use BMED to regenerate the MCFAs. And doing these experiments, we saw that we could quickly optimize them at the bench scale with really fast turnover times for our experiments and experimental results. And the sort of next steps that we are hoping for our BMED system is to remove CO2 from the feed solution to increase its efficiency. And of course, for our CO2 removal, what we showed was that using the same PDMS membranes that they use for inline extraction, we were able to achieve a really fast and easy method of CO2 removal. And we can remove, a lot of CO2 with just one PDMS membrane module. And currently, what Professor Lawson's team is doing is actually implementing the CO2 removal step in their bench scale bioprocess. Now, on top of BMED, we are also sort of exploring this other option where we would take our MCFA alkaline extract solution after, again, it's been accumulated and concentrated with MCFAs. What we are sort of looking at is if we were to manually acidify this alkaline solution with, let's say, hydrochloric acid down to a pH of 2, we could allow the phases to separate into both the MCFA oil and the aqueous phase. And then using BMED, what we can actually do is take this aqueous phase and run it through our BMED system to regenerate both acid and base, which can then be recycled back to this entire bioprocess. Now, what Deanna did actually was that she took this pH 9 alkaline solution, she did the manual acidification, and we were actually able to collect some of our MCFA oil product, as you can see here. Now, this MCFA oil product, we have sent that to one of our industry partners, CBS Bioplatforms, for further testing to see the purity of these MCFAs. And with that, I wanted to wrap up my presentation, and I would like to hand off the acknowledgements to Professor Lawson.
Okay, great work, everyone. Excellent presentations and overview of the technology. So we'd like to thank all of you for tuning in and are very excited to hear your questions. Before we get to that, I'd really want to make a big shout out to all of our industrial partners and who are in the Waste to Chemical Alliance. I think we have a very special group that really spans, folks on the technology side, folks on the waste handling side, as well as folks on the actual end users of the MCFA. So I'll just give a shout out again to them. That's Veolia, the Ontario Clean Water Agency, CBS Bioplatforms, the Ontario Water Consortium, and Vera. And I'd also like to give a big shout out to the City of Toronto, who's worked closely with us to access their facilities and obtain waste on sometimes, every couple of weeks. So they've been excellent in also providing feedback on our work. And most importantly, to all the students who've worked really tirelessly and put a lot of effort into getting this project off the ground. This project's been in swing now for just 2 years, and you can see with the presentations the amount of progress that's been made, and that's largely attributed to the students, some of which Diana, Jasmine, Renzo, and Ethan who presented today, but there's also many other students who have been involved in the project. So big shout out to all the hard work of the team. And with that, I'd like to open the floor to any questions. Thanks.
Yes, thank you very much, Chris, and congratulations to the team on all the progress that's been made, and congratulations on great presentation and a great session today. You can post your questions in the chat, or you can also raise your hand, but we do have a question here in the chat immediately from Ron Nielsen at MECP, and I'll read it out loud. Can a high-level approximate cost comparison be provided? How does the cost to produce MCFAs through these approaches compare to traditional MCFA production methods at commercial scale? Are these likely to be much more costly, somewhat more similar, or lower cost? So it's a multi-part question. I'm guessing that would be directed at you, Chris.
Yeah, great question. We've done some techno-economic modeling of the process, and we can say that the product can be produced competitively, certainly with existing processes. So we don't need special subsidies, anything like this to be competitively feel. Yeah. More importantly, the economic picture for making the MCFAs with anaerobic digestion versus, like, say, a biogas scenario also looks quite attractive from an economic perspective. Yeah.
Again, everybody, you can raise your hand either in the chat and ask any of the project team directly here, or you can post a question in the chat. We have another question from the participants here. Here we have one that's coming through the chat here again. That's from Kirby, City of London. Who are the end users for MCFAs? Are they widely spread? Thinking about selling the product?
Yeah, that's a great question. And so one of the unique things about our project that we've thought about from the onset was we actually have companies on our Waste to Chemical Consortium, one of them being CBS Bioplatforms, who is a major user of MCFAs and who have brought MCFAs to market in the animal feed space. And so they're very excited and interested in the results and in commercializing some of the bringing some of the MCFAs directly into their animal feed additives. So that's one of the main companies here in Canada is CBS Bioplatforms, but there are all other end users, Dow Chemical, BASF, other major chemical companies who work not only in the animal feed additive space, but also in solvent spaces and other end uses that are also interested in making MCFAs from more local sustainable sources. So I hope that answers your question.
Thanks, Chris.
Mm-hmm.
Don't have any further questions in the chat right now. And Kirby says thanks as well. Again, raise your hand if you have a question or write it in the chat or comment. I think we have another question in the chat. It's from Youngsek at Veolia. Could you tell us your scale up and commercialization plan or schedule?
Yeah, great question. So we are in the final year of this project, and our intent, the the results kind of have hit all the milestones that we've been anticipating or looking to achieve, primarily a yield of 0.3 gram MCFA per gram VS from the process. And so our next plan is to build a pilot plant and scale up the work. And so the pilot plant would provide an avenue for being able to produce a larger volume of the MCFAs to hand off to one of our partners, CBS Bioplatforms, for example, to further test and to stress test how the supply chain would actually look like. And so we're hoping to get that up and running in 2026 with a pilot plant. Yeah.
Excellent.
And of course, that would all be with the partners on some of the partners on our waste to chemical lines. Yeah.
Do we have any further questions from the audience? I'll wait a minute or so.
Yep. Lunchtime, people are getting hungry.
Yeah, I know. We do just a lot of congratulations. That's great. I'm glad everybody enjoyed the session and found it informative. Perhaps you were so great that you answered every question that anybody could have had.
There you go.
Or most of them, so that's very good. But we do have one more coming in here. And the question is, are you in any way partnering with any provincial ministry?
Yeah, that's a great question. We haven't yet, but this is something certainly as we move to the pilot plant, we'll be reaching out to provincial agencies to look at regulatory requirements, permitting. And so if anybody on this call is interested in this from one of those agencies, please feel free to reach out. It'd be great to loop you into the conversation, into the project.
I was going to say, I think we have a lot of participants from the Ontario government here, and I hear somebody who had called in. I can't see your name, but you've just unmuted yourself. Did you have a question?
Yes, it's Robin Skeets with the Ministry of the Environment. I just wanted to ask, is this something that is specific to food processing, or is it something that can also be done with municipal waste? And also with respect to size, what sort of, I know there are phase 1 and phase 2 anaerobic digesters, but is there a specific type of anaerobic digester required to do this process, whether it's just with the food and organic or just municipal wastewater?
Yeah, that's an excellent question. So the nice thing is, just like a typical anaerobic digestion process, it could be used for food waste, it could be used for wastewater biosolids, it could be used for dairy waste, it could be used for renewable biomass. So the wide mouthpiece of the digester still stays in place. And the unique thing about the technology is we can reroute the carbon to the MCFA product. So to answer the first part of your question, we could use biosolids, we could use other types of waste. The second part of your question, can you remind me what that was?
Yes, sizing. Is there a specific size of an anaerobic digester that's required? So, for instance, you're using the city of Toronto, that's pretty well the largest one probably in the country. But is there a certain size at which you can't go below?
That's a good question, and so that all ties into the economic piece. So the larger the facility, the economics play out better. That goes for any type of production facility. A lower limit on size, I don't know if I have an exact answer for you on that. I think we could certainly go lower though than what we've scoped out at, say, Disco or Dufferin. Yeah.
Thanks.
So it doesn't have to be a full massive city digester that we'd be retooling. And the nice thing about the technology is that it's actually quite modular. And so, if you have an existing facility, we feel it'd be relatively straightforward to retrofit the facility for the MCFA technology. So if you have a facility in mind and a size, certainly feel free to reach out, and we can talk about it more offline.
Well, it also poses one last question, if you don't mind, which is in terms of the length of time in the anaerobic digester. I mean, a lot of municipalities are looking for savings. Does it? Maybe I missed it, but does it spend more or less time, or the same amount of time in an anaerobic digester in terms of how your process works?
Yeah, great question. So it spends less time. So the retention time of the technology, this is unoptimized, but we probably need 12 days or less to actually optimize it. Moreover, we're using what's called a membrane bioreactor. And so we have a membrane that can separate the solid and hydraulic retention times of these systems. So that further reduces that tank volumes. And so we expect quite a significant reduction in footprint of the actual digester because of that. A normal digester is 20 days plus. So this could be seen also as a process intensification piece as well.
Thank you.
Thanks for that, Robin. I do see that we still have other MECP representatives here. You're welcome to comment in on Robin's comments as well, and whether you are interested in getting in touch with Chris or Jay or the entire team to see if there's collaboration opportunities on this. I mean, I think this is a good opportunity to do so. Not if there's any other questions from anybody else in the audience. We still have a fairly large audience in the room. Feel free to either put it in the chat or raise your hand, and the team will be happy to answer.
There is mud.
Right. I think it looks like, and there's a comment here from Adedoyan saying that I will encourage getting in touch with the Minister of MECP for participation. Oh, there's one more question here from Young Sek. He says, could you tell us about other competitors in this space and how your solution differentiates itself from theirs?
Yeah, that's a great question, Young. Thanks for that. So there's a couple existing companies that are also trying to scale up MCFA production from waste. And so one of them is Chaincraft, they're in the Netherlands, and then there's another one in the US called BioVeritas. I'd say those are kind of the two of the main companies. A main differentiator of our technology from theirs is they are really limited by more traditional open culture approaches. And so all the work that Ethan was presenting to you on the defined microbial consortia that we're developing and also being able to genetically modify is very unique to the capabilities of our lab. And actually some of those synthetic consortia that really are not even still remain unoptimized are already achieving higher MCFA yields than our optimized open community. So I think that is a very clear differentiator. Another differentiator is kind of the thought we put into the separation processes. And so Jay alluded to that a little bit, but having solvent-free membranes is a big important piece, especially if you're having a product go into an animal feed application, for example. And the energy savings with not having to go with distillation as well. So those are some of the really key differentiators. And again, with the synthetic microbiomes, one of the big areas of growth is not only just making the C6 and C8 MCFAs, but being able to make C10, C12, alcohols, alkanes, really this broad spectrum of oleochemicals that we're really now in a position to make with kind of our platform technology. Yeah. Ethan, that's right. Ethan is one of the differentiators, but really it's all the pieces coming together that really allow it to be differentiated. Yeah. Because we still need to put those microbes in the bioreactor.
Great. It looks like this would probably conclude our webinar today. Of course, if anybody has questions later on, you can feel free to reach out to the project team or the Ontario Water Consortium. We can always put you in touch with the project team. But thank you, everybody, for a wonderful webinar. Thank you to the project team and the execution of the webinar. And congratulations to the progress you've made on the project. We're excited to hear more as the time goes on. But I think this was a great success, and it was wonderful to hear from you, and it was wonderful for everybody to attend today. So thank you very much, everybody. Have a nice rest of your day.
Great. Thanks, everyone.
Thanks, Robert. Thanks, everyone.
Bye-bye.
Bye.
Thank you. Bye.
Thank you.
Thank you.